Coming to the sarcotubular system, it is highly specialized system of the internal conduction of depolarization within the muscle fiber. It is made up of T system, that is transverse tubular system, and a longitudinal sarcoplasmic reticulum, that is LSR. T system, they are inwardly directed extensions of the sarcolemma into the muscle fibers at the junction between A and I bands. Its function is the rapid transmission of action potential from cell membrane to all the fibrils in the muscle. The T-tubule protein is modified voltage-sensitive calcium channel known as dihydropyrimidine receptor, so named because it binds the class of drugs called as dihydropyrimidines. The main role of DHP receptor, however, is not to conduct calcium, rather to act as a voltage sensor. Longitudinal sarcoplasmic reticulum is longitudinally lies on the either side of the tubular system are found the sacs of the dilated LSR. These sacs are called as terminal cistern, which are rich in glycogen and calcium. Calcium channels in the sarcotubular reticulum are not voltage gated and allow the release of calcium into the cell. These are called ryanodine receptors. Transverse tubule with the two terminal cisterns are called as triad. There are two triad per sarcomere, and action potential on the T-tubule cause the DHP receptors to induce conformational change to open the ryanodine receptor channel. Calcium is thus released from the lateral sac of sarcoplasmic reticulum. This calcium binds with troponin, removes the blocking action of tropomyosin. So this is diagram showing the sarcoplasmic reticular system. This is the transverse tubules and these are longitudinal tubules. The transverse tubules helps in the conduction of action potential towards the longitudinal reticulum, where the action potential causes the activation of dihydropyrimidine receptors, which mechanically activate ryanodine receptors, and ultimately it leads to the release of calcium into the sarcoplasm, and this calcium is going to bind to the troponin, which is going to initiate the muscle contraction. The steps of excitation contraction coupling. The process by which depolarization of the muscle fiber initiates contraction is excitation contraction coupling. Sequence of events in excitation contraction coupling are the steps involved in the muscle contraction. So in the beginning there will be stimulation of the motor neuron that causes the release of acetylcholine into synaptic cleft, causing generation of end plate potential. Then there will be depolarization of the muscle membrane by increasing its permeability to sodium. Then there will be generation of action potential in the muscle fiber. Then there will be inward spread of action potential along the T system. Then spread of depolarization to the terminal system with the release of calcium in the myofibrils. Then there will be increase in the concentration of calcium in intracellular fluid by as much as 2000 times, which is going to lead to the muscle contraction. Role of calcium. Calcium binds with troponin C, causing tropomyosin to move laterally. This exposes the binding sites for myosin heads on the actin. It activates the prosthetic group of myosin filament, which acts as an enzyme, which is ATPase, myosin ATPase. It also helps in the steps in muscular relaxation. A few milliseconds after the action potential is over, sarcoplasmic reticulum begins to reaccumulate calcium. The calcium ions are actively pumped by calcium magnesium ATPase into longitudinal portion of the reticulum to terminal cistern for storage.